Rotavirus is the most common cause of severe diarrhea among infants and young children. It is a genus of double-stranded RNA virus in the family Reoviridae. Nearly every child in the world has been infected with rotavirus at least once by the age of 5. Immunity develops with each infection, so subsequent infections are less severe. Adults are rarely affected. There are 5 species of this virus, referred to as A, B, C, D, and E. Rotavirus A, the most common species, causes more than 90% of rotavirus infections in humans. The virus is transmitted by the faecal-oral route. It infects and damages the cells that line the small intestine and causes gastroenteritis. Although rotavirus was discovered in 1973 and accounts for up to 50% of hospitalizations for severe diarrhea in infants and children, its importance is still underestimated within the public health community, particularly in developing countries. In addition to its impact on human health, rotavirus also infects animals, and is a pathogen of livestock. Rotavirus is usually an easily managed disease of childhood, but worldwide more than 450,000 children under 5 years of age still die from rotavirus infection each year, most of whom live in developing countries, and almost 2 million more become severely ill. In the United States, before initiation of the rotavirus vaccination program, rotavirus caused about 2.7 million cases of severe gastroenteritis in children, almost 60,000 hospitalizations, and around 37 deaths each year. Public health campaigns to combat rotavirus focus on providing oral rehydration therapy for infected children and vaccination to prevent the disease. The incidence and severity of rotavirus infections has declined significantly in countries that have added rotavirus vaccine to their routine childhood immunization policies. Signs and symptoms: rotavirus gastroenteritis is a mild to severe disease characterized by vomiting, watery diarrhea, and low-grade fever. Once a child is infected by the virus, there is an incubation period of about 2 days before symptoms appear. Symptoms often start with vomiting followed by 4 to 8 days of profuse diarrhea. Dehydration is more common in rotavirus infection than in most of those caused by bacterial pathogens, and is the most common cause of death related to rotavirus infection. Rotavirus A infections can occur throughout life. The first usually produces symptoms, but subsequent infections are typically mild or asymptomatic, as the immune system provides some protection. Consequently, symptomatic infection rates are highest in children under 2 years of age and decrease progressively towards 45 years of age. Infection in newborn children, although common, is often associated with mild or asymptomatic disease. The most severe symptoms tend to occur in children 6 months to 2 years of age, the elderly, and those with compromised or absent immune system functions. Due to immunity acquired in childhood, most adults are not susceptible to rotavirus. Gastroenteritis in adults usually has a cause other than rotavirus, but asymptomatic infections in adults may maintain the transmission of infection in the community. Transmission: rotavirus is transmitted by the fecal-oral route, via contact with contaminated hands, surfaces and objects, and possibly by the respiratory route. The feces of an infected person can contain more than 10 trillion infectious particles per gram. Fewer than 100 of these are required to transmit infection to another person. Rotaviruses are stable in the environment and have been found in estuary samples at levels up to 1/5 infectious particles per use a gallon. The viruses survive between 9 and 19 days. Sanitary measures adequate for eliminating bacteria and parasites seem to be ineffective in control of rotavirus, as the incidence of rotavirus infection in countries with high and low health standards is similar. Disease mechanisms: the diarrhea is caused by multiple activities of the virus. Malabsorption occurs because of the destruction of gut cells called enterocytes. The toxic rotavirus protein NSP4 induces agent calcium ion-dependent chloride secretion, disrupts SGLT1 transporter-mediated reabsorption of water, apparently reduces activity of brush border membrane disaccharadases, and possibly activates the calcium ion-dependent secretory reflexes of the enteric nervous system. Healthy enterocytes secrete lactase into the small intestine. Milk intolerance due to lactase deficiency is a symptom of rotavirus infection, which can persist for weeks. A recurrence of mild diarrhea often follows the reintroduction of milk into the child's diet, due to bacterial fermentation of the disaccharide lactose in the gut. Diagnosis and detection. Diagnosis of infection with rotavirus normally follows diagnosis of gastroenteritis as the cause of severe diarrhea. Most children admitted to hospital with gastroenteritis are tested for Rotavirus A. Specific diagnosis of infection with Rotavirus A is made by finding the virus in the child's stool by enzyme immunoassay. There are several licensed test kits on the market which are sensitive, specific and detect all serotypes of Rotavirus A. Other methods, such as electron microscopy and PCR, are used in research laboratories. Reverse transcription polymerase chain reaction can detect and identify all species and serotypes of human rotavirus. Treatment and prognosis. Treatment of acute rotavirus infection is non-specific and involves management of symptoms and, most importantly, maintenance of hydration. If untreated, children can die from the resulting severe dehydration. Depending on the severity of diarrhea, treatment consists of oral rehydration, during which the child is given extra water to drink that contains small amounts of salt and sugar. In 2004, the WHO and UNICEF recommended the use of low osmolarity oral rehydration solution and zinc supplementation as a two-pronged treatment of acute diarrhea. Some infections are serious enough to warrant hospitalization where fluids are given by intravenous drip or nasogastric tube, and the child's electrolytes and blood sugar are monitored. Rotavirus infections rarely cause other complications and for a well-managed child the prognosis is excellent. Epidemiology. Rotavirus A, which accounts for more than 90% of rotavirus gastroenteritis in humans, is endemic worldwide. Each year rotavirus causes millions of cases of diarrhea in developing countries, almost 2 million resulting in hospitalization and an estimated 453,000 resulting in the death of a child younger than 5, 85% of whom live in developing countries. In the United States alone a year before initiation of the rotavirus vaccination program over 2.7 million cases of rotavirus gastroenteritis occurred annually. 60,000 children were hospitalized and around 37 died from the results of the infection. The major role of rotavirus in causing diarrhea is not widely recognized within the public health community, particularly in developing countries. Almost every child has been infected with rotavirus by age 5. It is the leading single cause of severe diarrhea among infants and children, being responsible for about 20% of cases, and accounts for 50% of the cases requiring hospitalization. Rotavirus causes 37% of deaths attributable to diarrhea and 5% of all deaths in children younger than 5. Boys are twice as likely as girls to be admitted to hospital. Rotavirus infections occur primarily during cool, dry seasons. The number attributable to food contamination is unknown. Outbreaks of Rotavirus A diarrhea are common among hospitalized infants, young children attending daycare centers, and elderly people in nursing homes. An outbreak caused by contaminated municipal water occurred in Colorado in 1981. During 2005, the largest recorded epidemic of diarrhea occurred in Nicaragua. This unusually large and severe outbreak was associated with mutations in the rotavirus A genome, possibly helping the virus escape the prevalent immunity in the population. A similar large outbreak occurred in Brazil in 1977. Rotavirus B, also called adult diarrhea rotavirus or ADRV, has caused major epidemics of severe diarrhea affecting thousands of people of all ages in China. These epidemics occurred as a result of sewage contamination of drinking water. Rotavirus B infections also occurred in India in 1998. The causative strain was named CAL. Unlike ADRV, the CAL strain is endemic. To date, epidemics caused by rotavirus B have been confined to mainland China, and surveys indicate a lack of immunity to this species in the United States. Rotavirus C has been associated with rare and sporadic cases of diarrhea in children, and small outbreaks have occurred in families. Prevention. Because improved sanitation does not decrease the prevalence of rotaviral disease, and the rate of hospitalizations remains high despite the use of oral rehydrating medicines, the primary public health intervention is vaccination. 2 vaccines against Rotavirus A infection are safe and effective in children, Rotarix by GlaxoSmithKline and Rota-TEQ by Merck. Both are taken orally and contain attenuated live virus. In 2009, the World Health Organization recommended that rotavirus vaccine be included in all national immunization programs. The incidence and severity of rotavirus infections has declined significantly in countries that have acted on this recommendation. In Mexico, which in 2006 was among the first countries in the world to introduce rotavirus vaccine, diarrheal disease death rates dropped during the 2009 rotavirus season by more than 65% among children aged 2 and under. In Nicaragua, which in 2006 became the first developing country to introduce a rotavirus vaccine, severe rotavirus infections were reduced by 40% and emergency room visits by half. In the United States, rotavirus vaccination since 2006 has led to drops in rotavirus-related hospitalizations by as much as 86%. The vaccines may also have prevented illness in non-vaccinated children by limiting the number of circulating infections. Rotavirus vaccines are licensed in more than 100 countries, but only 28 countries have introduced routine rotavirus vaccination. Following the introduction of routine rotavirus vaccination in the U.S. in 2006, the health burden of rotavirus gastroenteritis rapidly and dramatically reduced despite lower coverage levels compared to other routine infant immunizations. Clinical trials of the Rotarix rotavirus vaccine in South Africa and Malawi found that the vaccine significantly reduced severe diarrhea episodes caused by rotavirus, and that the infection was preventable by vaccination. Safety and efficacy trials of Rotarix and Rota-TEQ in Africa and Asia found that the vaccines dramatically reduced severe disease among infants in developing countries, where the majority of rotavirus deaths occur. A 2012 Cochrane review of 41 clinical trials that included 186,263 participants concluded Rotarix and Rota-TEQ are effective vaccines. Additional rotavirus vaccines are under development. In September 2013, the vaccine will be offered to all children in the UK, aged between 2 and 3 months, and it is expected to halve the cases of severe infection and reduce the number of children admitted to hospital because of the infection by 70%. International non-governmental organization PATH, the WHO, the U.S. Centers for Disease Control and Prevention, and the GAVI Alliance are working to bring rotavirus vaccines to developing countries, where children face the greatest burden. Through the Rotavirus Vaccine Program and the Accelerating Vaccine Introduction Initiative, these groups are partnering with research institutions and governments to reduce child morbidity and mortality from diarrheal disease by making a vaccine against rotavirus available for use in developing countries. Infections of other animals. Rotaviruses infect the young of many species of animals and they are a major cause of diarrhea in wild and reared animals worldwide. As a pathogen of livestock, notably in young calves and piglets, rotaviruses cause economic loss to farmers because of costs of treatment associated with high morbidity and mortality rates. These rotaviruses are a potential reservoir for genetic exchange with human rotaviruses. There is evidence that animal rotaviruses can infect humans either by direct transmission of the virus or by contributing one or several RNA segments to reassortance with human strains. Virology, types of rotavirus. There are 5 species of rotavirus, referred to as A, B, C, D and E. Humans are primarily infected by species A, B and C, most commonly by species A. All 5 species cause disease in other animals. Within rotavirus A there are different strains, called serotypes. As with influenza virus, a dual classification system is used based on 2 proteins on the surface of the virus. The glycoprotein VP7 defines the G serotypes and the protease-sensitive protein VP4 defines P serotypes. Because the 2 genes that determine G types and P types can be passed on separately to progeny viruses, different combinations are found. Structure. The genome of rotavirus consists of 11 unique double helix molecules of RNA, which are 18,555 nucleotides in total. Each helix, or segment, is a gene, numbered 1 to 11 by decreasing size. Each gene codes for one protein, except genes 9, which codes for 2. The RNA is surrounded by a 3-layered icosahedral protein capsid. Viral particles are up to 76.5 nm in diameter and are not enveloped. Proteins: there are 6 viral proteins that form the virus particle. These structural proteins are called VP1, VP2, VP3, VP4, VP6 and VP7. In addition to the VPs, there are 6 non-structural proteins that are only produced in cells infected by rotavirus. These are called NSP1, NSP2, NSP3, NSP4, NSP5 and NSP6. At least 6 of the 12 proteins encoded by the rotavirus genome bind RNA. The role of these proteins play in rotavirus replication is not entirely understood. Their functions are thought to be related to RNA synthesis and packaging in the virion, mRNA transport to the site of genome replication, and mRNA translation and regulation of gene expression. Structural proteins: VP1 is located in the core of the virus particle and is an RNA polymerase enzyme. In an infected cell, this enzyme produces mRNA transcripts for the synthesis of viral proteins and produces copies of the rotavirus genome RNA segments for newly produced virus particles. VP2 forms the core layer of the virion and binds the RNA genome. VP3 is part of the inner core of the virion and is an enzyme called guanylyl transferase. This is a capping enzyme that catalyzes the formation of the 5' cap and the post-transcriptional modification of mRNA. The cap stabilizes viral mRNA by protecting it from nucleic acid-degrading enzymes called nucleases. VP4 is on the surface of the virion that protrudes as a spike. It binds to molecules on the surface of cells called receptors and drives the entry of the virus into the cell. VP4 has to be modified by the protease enzyme trypsin, which is found in the gut, into VP5 and VP8 before the virus is infectious. VP4 determines how virulent the virus is and it determines the P-type of the virus. VP6 forms the bulk of the capsid. It is highly antigenic and can be used to identify rotavirus species. This protein is used in laboratory tests for Rotavirus A infections. VP7 is a glycoprotein that forms the outer surface of the virion. Apart from its structural functions, it determines the G-type of the strain and, along with VP4, is involved in immunity to infection. Non-structural viral proteins: NSP1, the product of gene 5, is a non-structural RNA-binding protein. NSP2 is an RNA-binding protein that accumulates in cytoplasmic inclusions and is required for genome replication. NSP3 is bound to viral mRNAs in infected cells and it is responsible for the shutdown of cellular protein synthesis. NSP4 is a viral enterotoxin to induce diarrhea and was the first viral enterotoxin discovered. NSP5 is encoded by genome segment 11 of Rotavirus A and in virus-infected cells NSP5 accumulates in the viroplasm. NSP6 is a nucleic acid-binding protein, and is encoded by gene 11 from an out-of-phase open reading frame. This table is based on the simian rotavirus strain SA11. RNA protein coding assignments differ in some strains. Replication: rotaviruses replicate mainly in the gut, and infect enterocytes of the villi of the small intestine, leading to structural and functional changes of the epithelium. The triple protein coats make them resistant to the acidic pH of the stomach and the digestive enzymes in the gut. The virus enters cells by receptor-mediated endocytosis and form a vesicle known as an endosome. Proteins in the third layer disrupt the membrane of the endosome, creating a difference in the calcium concentration. This causes the breakdown of VP7 trimers into single protein subunits, leaving the VP2 and VP6 protein coats around the viral dsRNA, forming a double-layered particle. The 11 dsRNA strands remain within the protection of the two protein shells, and the viral RNA-dependent RNA polymerase creates mRNA transcripts of the double-stranded viral genome. By remaining in the core, the viral RNA evades innate host immune responses called RNA interference that are triggered by the presence of double-stranded RNA. During the infection, rotavirus produces mRNA for both protein biosynthesis and gene replication. Most of the rotavirus proteins accumulate in viroplasm, where the RNA is replicated and the DLPs are assembled. Viroplasm is formed around the cell nucleus as early as 2 hours after virus infection, and consists of viral factors thought to be made by two viral non-structural proteins, NSP5 and NSP2. Inhibition of NSP5 by RNA interference results in a sharp decrease in rotavirus replication. The DLPs migrate to the endoplasmic reticulum where they obtain their third, outer layer. The progeny viruses are released from the cell by lysis. History. In 1943, Jacob Light and Horace Hodes proved that a filterable agent in the feces of children with infectious diarrhea also caused scours in cattle. 3 decades later, preserved samples of the agent were shown to be rotavirus. In the intervening years, a virus in mice was shown to be related to the virus causing scours. In 1973, Ruth Bishop and colleagues described related viruses found in children with gastroenteritis. In 1974, Thomas Henry Flewitt suggested the name rotavirus after observing that, when viewed through an electron microscope, a rotavirus particle looks like a wheel. The name was officially recognized by the International Committee on Taxonomy of Viruses 4 years later. In 1976, related viruses were described in several other species of animals. These viruses, all causing acute gastroenteritis, were recognized as a collective pathogen affecting humans and animals worldwide. Rotavirus serotypes were first described in 1980, and in the following year, rotavirus from humans was first grown in cell cultures derived from monkey kidneys, by adding trypsin to the culture medium. The ability to grow rotavirus in culture accelerated the pace of research, and by the mid-1980s the first candidate vaccines were being evaluated. In 1998, a rotavirus vaccine was licensed for use in the United States. Clinical trials in the United States, Finland, and Venezuela had found it to be 80 to 100% effective at preventing severe diarrhea caused by Rotavirus A, and researchers had detected no statistically significant serious adverse effects. The manufacturer, however, withdrew it from the market in 1999, after it was discovered that the vaccine may have contributed to an increased risk for intussusception, a type of bowel obstruction, in 1 of every 12,000 vaccinated infants. The experience provoked intense debate about the relative risks and benefits of a rotavirus vaccine. In 2006, 2 new vaccines against rotavirus A infection were shown to be safe and effective in children, and in June 2009 the World Health Organization recommended that rotavirus vaccination be included in all national immunization programs to provide protection against this virus. References. External links. WHO Rotavirus webpage, Rotavirus on Centers for Disease Control and Prevention site, Virals Rotavirus, Vaccine Resource Library, Rotavirus, defeatdd.org, Centers for Disease Control and Prevention. CH18, Rotavirus. In Atkinson W., Wolf S., Hambisky J. Epidemiology and Prevention of Vaccine-Preventable Diseases. Washington, D.C., Public Health Foundation PPA 263 Euro 274 3D macromolecular structures of rotaviruses from the M-Data Bank, EMDB, Ramig, R.F. Pathogenesis of intestinal and systemic rotavirus infection. Journal of Virology 78, 10213 Euro 20 doi, 10.1128/JVI. 78.19.10213-10220.2004. PMCA 516399. PMIDA 15,367,586A.